Very good morning, my dear friends. So today's eminent resource person, Dr. K. Madhuri Madam Garu, Assistant Professor, Department of Biology and Biotechnology, one of the prestigious university in our Telangana, that is Mahatma University, Nalgonda. Madam is having the lots of duties, but for our Madam has given a wonderful time for us. So better utilize this wonderful opportunity. Madam is going to explain today's our class, that is replication of DNA. Madam, please let you start this session, madam.
Sure, sir.
And share your screen also.
Sure, sir. Sure, sir. Yeah. Very good afternoon to one and all. I'm Dr. Madhuri from Department of Biotechnology, Mahatma Gandhi University. Thank you very much, Tirupati sir, for giving me this opportunity. Last class, I have just started. I gave a brief session on the structure of DNA, where we have come across the basic structure between the difference between the DNA as well as the RNA. And we have gone through the Watson and Crick model of DNA. So today I wanted to continue the session with DNA replication, where we will deal mostly with the types of DNA replication, where I will have a touch with, I'll go in depth of what is semi-conservative model of DNA replication. Then we will also see the difference of the prokaryotic DNA replication, eukaryotic DNA replication, as well as the inhibitors of DNA replication. So this I would like to cover. And as you can see in our next slide, we very well know that the DNA is heritable, like it is transferred from one generation to another generation, where the information present in this DNA is passed on to the next generation, and that's how it is carried forward. So how it is carried forward? So it undergoes replication, then only the information is carried forward. So central dogma, the information which is transformed by replication, by transcription and translation, is called as central dogma. That is from DNA replication to transcribed to RNA and then translated to a protein. So this is called as central dogma, which every student should have an knowledge on it. So as we can see that the DNA replication, what is replication? It's nothing but doubling. Okay, where the DNA replication is a biological process that occurs in all living organisms and copies their exact DNA. It is the basis for biological inheritance. As I mentioned you, it is inherited. The information has been inherited from one generation to another generation by replication process. So this replication is nothing but synthesis of daughter DNA from parental DNA by the enzyme DNA polymerase. So there are n number of enzymes which are involved in this replication process. So once I give you the tinge of DNA replication, I'll show you the animation also, how the enzymes are going to attach to the DNA and the DNA is being doubled and so on. So as you can see, the simple funda or the formula for the replication is your DNMP 2N plus your DNTP, which gives rise to the lengthened DNA. That is your DNMP is nothing but deoxyribonucleoside monophosphate. So these monophosphate in N, that is indefinite, okay, where they attach, and your deoxyribonucleotide triphosphate gives rise to your DNMP and plus 1 plus your PPI. Your PPI represent your pyrophosphate. So no confusion here. This is the formula for the simple DNA to go for the lengthened DNA. So as you have already seen in the DNA structure, we mentioned last time that your GC, ATGC bondings, right? So 5 prime to 3 prime, 3 prime to 3 prime to 5 prime, you have two DNA strands where they run parallel to each other, and always G binds with C and A binds with T. AT-GC binding we know. So these two DNA strands, that is the parental DNA strands, they unwind in DNA replication, where each strand will be acting as a parental strand, and to this strand a new DNA, that is the green color you can see, that is the daughter strand forming. So this is in simple the DNA replication. So as you can see, a reaction in which the daughter DNAs are synthesized using the parental DNAs as the template. Template is what? Template is a kind of a copy. Like we are trying to take a standard copy to make another copies. So that we take it as a template. So transferring the genetic information to the descendant generation with high fidelity. Like without fidelity is like live. Okay. So high fidelity is called as DNA replication. See, as you can see that you can see the parental DNA, which is in red color, right? So this parental DNA, they try to separate and give rise to your daughter DNA, which are in yellow color. So as you can see that, yeah. Um Yeah, so you can see the parental DNA is replicating and giving rise to two new daughter DNA strands. So yeah, there are three possible replication patterns, that is semi-conservative replication, conservative replication, and dispersive replication. So generally the replication occurs in three forms: semi-conservative, conservative, and dispersive. But generally you can see that in semi-conservative, that is the original DNA, that is the parental DNA, is forming, like they separate, and from each parental there is a new daughter strand form. Till now in this fidelity which we were discussing, yeah, this kind of replication is a semi-conservative replication. In conservative replication, what is happening? There won't be any separation of the parental DNA, but from the parental DNA, a totally new daughter DNA will be produced. Whereas in dispersive, you can see that the parental DNA, in between you can see the new strand formation, that is the red color in between of the blue color. This is dispersive replication. So majorly the higher end of organism, they go for semi-conservative method of replication. So as you can see that each parent strand serves as a template, like a standard copy for new strand, and the two new DNA strand each have one old as well as one new strand. So in this way, the semi-conservative mode of replication takes place. So as we can see that in what are the characteristics of replication? So as we see in semi-conservative, most of the organism undergo semi-conservative replication, and this is bidirectional. Both the directions, the replication takes place, and it is semi-continuous mode of replication. As I mentioned, it is highly stable, high fidelity. So you can see this was demonstrated by Messelson and Stahl experiment in 1958, that the organisms undergo semi-conservative mode of replication. So this experiment was in boom, like it was highly adopted, and we still go for many experiment based on these Messelson's and Stahl's experiment. So here what they have done is they have extracted the DNA and centrifuged to equilibrium in cesium chloride density gradients. In simple, I'll explain you students, where they have taken the DNA by using a isotope of nitrogen, a heavy isotope of nitrogen, that is N15. This was there in original parent molecule, and then it was incorporated with the DNA, with the liquid containing the nitrogen 14 isotope. So what they have seen is this experiment more clearly I can explain you in a petri dish. So here they have taken the nitrogen 15 isotope. E. coli was grown in the presence of a heavy isotope, that is nitrogen 50, for many generations, where the parent was incorporated with this N15 nitrogen. Then what did they do? They plated in the media containing N14. You can see in the second generation, that is in this plate, you can see nitrogen 14, where the E. coli placed in media containing N14, a light isotope of nitrogen here. So what is happening then? This two, when they were recombined or when they were combined, what happened? The cells were broken up to extract the DNA, and when the DNA was suspended in cesium chloride, because it's a density matter, so they have chosen the cesium chloride, where the density gradient centrifuge, where you can see the lower cesium concentration and high cesium concentrate. So here you can see both the strands with heavy nitrogen 15, and in F1 generation, they have seen the DNA with one heavy isotope, that is N15, and one with light isotope, that is N14. Whereas in F2 generation, they have seen two light strands and one heavy strand and one light strand DNA. So this mechanism, this method has confirmed that the DNA undergoes semi-conservative method of replication. So in the sample 1, 0 minutes, in the sample after 20 minutes, you can see the first generation. So in the first, in this particular one, you can see N14 DNA, and here you can see the N14, N15 DNA, and the last you can see the N15 DNA. So this Michelson and Stahl experiments had made clear that the replication is by semi-conservative. You can see here in the 3 rounds of replication, this is the original DNA with blue color. First round, you can see one red, that is the daughter DNA formed. And in the second one, even the daughter will become the parent here, right? So that's why you can see two reds again, that is two new daughter. And in the third round, you can see but still the parent is been continuing even in the third round, the blue one. So in 16 minutes, these are the generations. I mean the 3 rounds of replication, and this confirms a semi-conservative method of replication. So now you can see that how the DNA unwinds. So next step, we will see the unwinding of the DNA, and as we have seen the DNTPs, that is deoxyribonucleic triphosphates and monophosphates, how they attach to the DNA for forming the new DNA strands. So you can see the 5', 3' DNA getting unwind here, and here it is in the form of a fork shape. That's why it is called as replication fork formation. A fork which we use to eat, right? The fork. Yeah. So you can see that from 5' to 3', if you have CACTGGTACATACCT, and in your 3' you have the opposite one, your amino acids, that is GTGA, So these are the parental one. And as you can see that when they get separated, the new strand is having the opposite, that as you can see CGAT, and for this the same one. So identical base sequences you can see in the new strand formation. And as we go on in the semi-conservative, half of the parental DNA molecule is conserved. What do you mean by conserve? Preserve, right? So it is the parental molecules are conserved in each new double helix paired with the newly synthesized complementary strand. So this is called as semi-conservative replication. So you can see that the parental DNA molecule, whatever is being passed on to the next generation, it is conserved, and even that is involved in the formation of new double helical structure, and to which the complementary strand is formed. So here, once again, we repeat of the semi-conservative replication. Half of the parental DNA molecule is conserved in each new double helix paired with a newly synthesized complementary strand. This is called semi-conservative mode of replication. So as we mentioned, the direction is bidirectional. Okay, we already mentioned the characteristics of the semi-conservative method of replication. So you can see the direction of the DNA. It is in the ori region where the direction starts. So as you can see, first it starts unidirectionally. Then as the fork opens, you can see the replication is bidirectional. So let us see this mechanism, bidirectional replication. We already mentioned that the replication starts by unwinding the double-stranded DNA. We mentioned that the DNA gets unwind, right? So where it is getting unwind, it gets unwind at a particular point. And that particular point is called as ori site or origin site. The origin site, in shortcut, we call it as ori site, followed by the synthesis on each strand. So the parental double-stranded DNA and two newly formed double-stranded DNA form a Y-shaped structure called the replication fork, which we already mentioned. So as you can see that, so this is the original DNA. So in this original DNA, there will be certain sites called as the origin site or the ori site where the replication starts and where the unwinding of the DNA starts. So as the DNA gets unwind, you have two strands left over. The blue one is the parental strands. Okay. So from the blue one, the new strands which are in the pink color are synthesized. So the strand which is running from 3' to 5' end is the leading strand. Students, most of the students get confused between the leading strand and the lagging strand. So the leading strand will have the replication, like the synthesis of the daughter strand without any obligations or without any hurdles. So that's why it is a leading strand. And as you can see, it starts from 3' to 5'. Whereas the lagging strand is generally the strand where you have the breakup of the daughter strand form. That's why it is called lagging. There is a lagging. There is a kind of deficiency in this particular strand. That's why we call it as lagging, because it doesn't have the lagging strand has a deficiency to form a proper daughter strand, new strand to be formed without any obligations. So that's why it is lagging strand. So as you can see, the direction of the movement of the fork, and in the lagging strand, as you have the deficiency, that's why the Okazaki fragments are formed. We will see in depth of it so that we will not have any confusion between the leading strand as well as the lagging strand. So the one which is leading for the formation of new daughter strand without any obstacles is the leading strand. Okay, so try to remember in that way. So as you can see, the replication in prokaryotes, this is the original picture, original electron microscopic slide, which is showing the replication of the in prokaryotes, and the replication process starts from the ori region, that is the origin, and proceeds into opposite direction, and it is named as a theta replication. It's a symbol of theta. So this is called as a theta replication. So as you can see, now before going to replication, we should also know the superheroes involved in replication, that is the enzymes. They do not get any change, they don't get changed, but they are very much important to perform a replication. So let us see the replication enzymes and the proteins involved in DNA replication. So the first important enzyme is DNA polymerase. This is the enzyme which matches the correct nucleotides, like with A, the T, with G, the C. So this particular matching is possible only by the DNA polymerase enzyme, where it matches the correct nucleotides, then joins, polymerizes adjacent nucleotides to each other. You know very well that poly, like a continuous chain, is formed. Next comes the helicase. Now we very well said that the DNA has to get unwinded. So how is this unwinding possible? There is a motor, there's a machine which helps for unwinding of the DNA, and that is the helicase enzyme. So this helps in unwinding of the DNA and melts it. Next comes the primase enzyme. This primase provides the RNA primer to start the polymerization. So for DNA synthesis to occur, it has to be initiated by a primer, a sequence of RNA. That sequence is called as RNA primer. So as you can see, those are the enzymes. Major enzymes are polymerase, DNA polymerase, helicase, primase. Then comes the single strand binding proteins. Now, these proteins help the DNA single stranded after it has been melted by helicase so that they will not get joined again. So these proteins helps the DNA to be away from each other, to be single for a little longer period of time. And here comes the gyrase enzymes. This is a topoisomerase, and there are a number of topoisomerase enzymes. One of the enzyme is the gyrase enzyme, and this enzymes that relieves torsional strain in the DNA molecule so that it can complete the replication. As you can see, the ligase enzymes, this joins the adjacent DNA strands together. We mentioned that in the lagging strand, there are where there is no continuous attachment of these nucleotides, there is a formation of Okazaki fragments. I will also deal with Okazaki, okay? And these fragments are joined with the help of these ligase enzyme. Now we mentioned RNA primer. The RNA primer after replication, it has to leave. So those gaps are filled and the DNA is joined with the help of the ligase enzyme. And as you can see, the telomerase, this finishes the end of DNA strands in eukaryotes. Like once the replication ends, it has to come up to an endpoint. So this endpoint is basically finished by the telomerase enzymes. So these are the enzymes. Just revise: DNA polymerase, helicase, primase, gyrase, ligase, and telomerase enzymes. Now let us see how the DNA replication starts. So as we mentioned, the DNA protein, which helps in the recognition of the ORI sequence. It is around the molecular weight of 50,000 and it has a subunit of 1. And this DNA protein is used for recognizing of the ORI sequence. This table will give you a grand picture of the proteins and the enzymes involved in it. As you can see, the DNA B protein, that is the DNA helicase enzyme, and this has 6 subunits. This helps in opening of the double-stranded DNA, unwinding of the double-stranded DNA. Next comes DNA C protein. This helps in, it assists the DNA B to bind at ORI site. So this B is binded to the ORI site with the help of C protein. A recognize, B goes and unwinds, and C protein helps the binding of the helicase enzyme. And your DNA polymerase, as we mentioned, this helps in synthesis of the new DNA strands by attaching your NTPs. So as you can see here, NMPs. So as you can see here, the DNA G protein, this is your primase, and this synthesizes the RNA primer. Primase, you can remember in that way, the primase helps in synthesis of RNA primer, which is triggering or initiating the process. As you can see that single strand binding proteins, they play a very important role. They bind a single stranded DNA and helps to maintain the stability of the DNA in single strand form. And of course, you can see the gyrase, which relieves the torsional strain generated by unwinding. So as you can see that once the helicase comes and binds this particular gyrase, it is helping the strain to unwind the strain. What I should say, it helps in acting of the twisting or the state of being twisted, especially the twisting of the cut. Okay, so this torsion has been relieved by the gyrase enzyme. So as you can see, the DNA polymerases of prokaryotes, you have DNA polymerase 1. This was the first DNA polymerase, DNA dependent DNA polymerase, and this was discovered by Arthur Kornberg. He's a scientist. The right side is the original picture of Arthur Kornberg, who received the Nobel Prize. And he's a person who has identified the DNA polymerase enzyme, and the DNA polymerase is considered as Kornberg enzyme after the discovery of Arthur Kornberg. So as you can see later on, I think you have an idea, but still I'll just tell you, we have three types of DNA polymerases. And the first one is DNA polymerase 1, which was identified by Kornberg. Later on, they have identified DNA polymerase 2, DNA polymerase 3. In short, we call it as DNA pol 1, pol 2, pol 3. And these were identified later. And all of them have the following biological activity. What is their activity? They run from 5' to 3' polymerase activity and exonuclease activity. So as you can see that if you compare the DNA polymerases of E. coli, you can see the 1, 2, and 3. These are pol A, pol B, pol C. And you can see that they help an exonuclease, a biological activity which we mentioned. And the gene structure is pol A, pol B, and pol C. This is DNA of E. coli. That's why DNA E. Okay, let's not go in depth of this particular. If you want, I will share this slide share with sir. You can take from him, and later on you can do the comparative analysis of these three enzymes. So you can see that this exonuclease function, like what is this activity? If you see now, this 5' to 3', it removes the primer or excites the mutated segment. And in 3' to 5' exonuclease activity, they excise the mismatched nucleotide. Excise is removal. So they remove. Now, you know very well the G has to go and bind with C, not with A. So this excision, this kind of removal of this nucleotide is performed by these enzymes. So biological activity of these enzymes. If there is any mismatch, if there is any mutation, what is mutation? A sudden change in the sequence. So this can be repaired, or this can be removed. This can be excised by this particular enzyme. So as you can see, the DNA polymerase 1, this mainly is responsible for proofreading and filling the gaps and repairing the DNA damage. So if there is like ATGC, any sequential change, so this particular enzyme helps to remove and correct. So proofreading. Proofreading is correctness. It goes for editing, proofreading, and filling the gaps and repairing the DNA damage also. So that's why in my first slide, I mentioned the replication with DNA damage too. So as you can see that this DNA polymerase, it will be cleave the, as you can see, it will be cleaving the sequences in small fragments and large fragments, and it helps in the exonuclease activity. So this we can study further when we do the DNA damage mechanism. Whereas coming to DNA polymerase 2, this is having a temporarily functional when DNA polymerase 1 and 3 are not functional. So this DNA polymerase, still it is having the capability for doing synthesis on damaged template. And this participates in DNA repair mechanism. Whereas 3, this is heterodimer enzyme, and it is composed of 10 different subunits. And this each polymerase 3, they are having the highest polymerization activity and the true enzyme responsible for elongation process. So this helps in attachment of your DNT piece. So as you can see that this particular enzyme subunits of polymerase 3, as you can see, it is having a subunits of 10, and this is the core enzyme, helps in elongation of the the DNA. So this is the structure of DNA polymerase 3. You have an alpha, then you have a theta and an epsilon, and you can see that this structures of DNA polymerase, helicase, gyrase, and all I would like to take in the next episode. since we have entered into this DNA replication, let's rush to the replication part of the DNA. Okay. This is a enzyme which is trying to catch hold of the DNA, and this helps in unwinding of the DNA. Okay. So here comes the helicase. This is the helicase enzyme. This helicase enzyme helps the DNA to unwind. So before going to the unwinding of the DNA, I want to show you a video where the DNA is getting unwind, where the helicase is coming into the picture and holding the DNA replication. A small one, so that it will be easy for us to understand. So in this, you can see the DNA molecule is made up of two double-stranded, and you can see the twisted form of the DNA. Now each DNA is getting unwind, and you can see the ACGT letters over here. Okay, and you can see the two strands are complementary to each other, and they will be in A in the opposite strand, and whereas C in the other. You can see the ATGC. Okay, and each strand has 5' end and a 3' end. The opposite is from 5' to 3' is 3' to 5'. So the two strands run in opposite direction. So this determines how each strand of DNA is replicated. And you can see the first step in DNA replication is separation of the DNA strand. So this is the enzyme. I just wanted to show you the enzyme helicase, how this enzyme is unzipping the DNA. You can see that the 5' to 3', 3' to 5' DNA has been unzipped with the helicase enzyme. So you can see once it is unzipping the helicase, the DNA with the help of the helicase enzyme, you can see the replication fork. It's in the form of a fork, right? Which we use to eat the fruits and all. The spoon with the sharp ends is a fork, right? So that's how the replication fork is formed. And as you can see, the separate strands each provide a template for creating new strands. So this acts as a parental strand for the formation of the new strand. So here comes, once they separate the parental strand, the template, they are called the template strand. Now the enzyme primase comes into the picture. So this enzyme called primase start the process, where it will start the replication at the ori region by initiating the attachment of RNA primers. So here comes a primase. It has attached to the ori region, and you can see the RNA bases coming and attaching, and this enzyme makes a small piece of RNA, that is your RNA primer. Okay, where they attach at the ori region, like they are the kind of starting points where the RNA attaches, a primer attaches and initiates the RNA replications. So this marks the starting point for the construction of new strand of DNA, let it be in prokaryotes or in eukaryotes. Okay, so as you can see, next the enzyme DNA polymerase. So we have mentioned three types of polymerase: DNA polymerase 1, 2, and 3. So here comes a DNA polymerase which binds to the primer, and next it starts synthesizing the DNA by strands, where the new DNA bases will come and attach the old template from 5' end to 3' end. You can see these small particles. These are the NTPs, the deoxyribonucleotide monophosphates, which will come and attach to your new strands, forming your triphosphate. So one of the new strands of DNA, the leading strand, is made continuously, and the DNA polymerase is adding the bases one by one in the 5' to 3' direction. So that is the leading strand. The leading strand do not have any obstacles, and continuously the replication is starting. Whereas The other strand is the lagging strand. Okay. And this strand, because it runs in the opposite, as you can see, this lagging strand, they